Hello guys, this is Anna, and today I'm going to discuss some contraction mechanisms. Specifically, I'm going to start with the skeletal muscle contraction. You know that the skeletal muscle is attached to the bone with the help of the tough connective tissue called tendon. Here's our tendon, and when the muscle actually contracts, it places some tension on the tendons and helps our skeleton to move. Well, during the contraction, the angle at the joint actually changes. And if the angle decreases, this is called the flexion movement. If the angle increases, this is called the extension movement. Also, you have to know that there is a difference between agonist and antagonist muscles. Well, the prime mover of any skeletal movement is called an agonist muscle. To put it simply, I can tell you a really nice example. Well, in flexion, the flexor muscle is an agonist, but the extensor muscle would be antagonist because actually flexor and extensor muscles do the opposite things. And for example, when we are doing some like really complex movement, only agonist muscles have to be turned on and antagonist muscles have to be turned off, in order to get regulated complex movement. And this is, of course, regulated by the brain, by the CNS, by nerves in our body, and so on. Okay, let's take a look at the skeletal muscle fiber. Well, the muscle fiber actually is composed of numerous myofibrils. These are the myofibrils, and the myofibrils are made up of myofilaments, which are represented by the actin and myosin. Then we also have some sarcoplasm, which is the cytoplasm of muscle fiber, and sarcolemma, which is the plasma membrane, actually, of the muscle fiber. And also I have to add that each skeletal muscle fiber is multinucleated, and here you see the nuclei, which are actually multiple instead of one or two or even three. Now let me take a look at this picture. You know that I've mentioned that actually nerves control our muscle movement, and specifically our somatic motor neurons control the muscles because somatic nervous system actually obeys us and is responsible for some voluntary movements. And here you see that we have some motor units inside the muscle. And specific motor units are responsible for contraction of only specific area of the muscle. Well, the other area is controlled by the other unit. And here the motor unit is kind of zoomed in, and you see that the neurons are branched and they are specifically attached to certain fibrils, and only those myofibrils are controlled by them. Other ones would be innervated by the other neurons, okay? So just imagine that somatic motor units actually are dispersed in the whole, along the whole length of the muscle, okay? Okay, now let's talk about the specific structure called sarcomere, which is a basic unit of the muscle fiber, and which is actually responsible for this whole contraction thing. Well, it's made up of thin and thick filaments. The thin filaments are actin and some associated proteins, which I'm going to discuss a bit later. And thick filaments are represented by myosin. Well, myosin filaments slide past actin, and these actually leads to contraction. Well, here I have to mention some examples of thin and thick filament. Well, I mentioned that myosin is an example of thick filament, but besides actin, some additional proteins are troponin and tropomyosin, which are also part and representatives of thin filaments. This is our whole structure of myosin, the myosin heads, the actin. Additionally, we have some troponin molecules there, which are not seen right now. But before actually describing them, let me move on to the next part, the next slide, which actually describes different areas of the whole sarcomere. And the sarcomere is the area located between the two Z discs. Here you see in the middle the myosin myosin chains, which are actually thick filaments, and actins, little violet actins, which are thin filaments. Yeah. Now let's take a look. Here we have the I band. Where is it? I band, which only includes the actin chains, only actin molecules. The H band, which only includes the myosin chains, yeah? But as for the A band, well, A band includes the both myosin and overlapping actin. When the contraction happens, well, these myosin and actin actually like past each other, slide past each other, which means that the structure of the sarcomere changes a bit. For example, in the central area, we don't have the area free of some actin molecules, because now all the actin and myosin molecules are overlapped, and we don't have the area free of myosin molecules at the end of the sarcomere. Yeah, so that's why, since both of them are now overlapping, only thing that does not change, only band that doesn't change is A band, which includes all the lengths of the myosin molecules, and now includes the overlapped actin molecules as well. Yeah, but the I band here and the H band in the middle both have disappeared during contraction process. Okay. Now, this slide specifically shows the content of each band, and they are like indicated with different colors just to make easier for you to memorize. So make sure you come back to this slide and take a look at it. Now let me move on to troponin. Well, I've mentioned that troponin is part of the thin filaments, yeah? And turns out that we have three types of troponins. Troponin C, which is responsible for calcium binding, troponin T, which actually binds to tropomyosin. This is another protein also represented through thin filaments, and troponin I, which inhibits the myosin binding to actin. And what is an interesting thing is that when some calcium arrives into the cell, calcium can bind to the troponin. You remember there is troponin C, which is specifically for calcium binding. Yeah, when calcium binds to troponin and this complex is produced, it leads to removal of the tropomyosin from the myosin, and actually the binding site for the actin is now free. For you to imagine it better, let me open the video, a short video. I'm going to turn off the voice. This is our whole sarcomere. And let me move on to this part specifically. Well, here, as you see, this is the chain of actin, and this is our myosin head. The whole myosin chain and the myosin head is located there. Tropomyosin actually blocks the binding areas in order to prevent the attachment of myosin head to actin, and little troponin molecules are also attached to tropomyosin. These are troponin C, troponin I, and troponin T. Yeah, troponin T is attached directly to tropomyosin. Troponin I is another molecule inhibiting these binding areas, and troponin C is ready to attach to calcium when it's available inside the cell. Now let's imagine that this is our calcium. Well, when the amount of the calcium actually increases inside the cell, the calcium molecules bind to, calcium molecules actually bind to troponin C. Then troponin I changes its conformation, and tropomyosin now becomes moved away from the binding sites. Here are our binding sites for the myosin head, so that the myosin is now able to attach to actin, and this picture shows you where it actually attaches. Afterwards, certain things have to happen inside the muscle, and I'm going to explain them right now. Well, now you already know how myosin head can actually attach to the actin, yeah? But afterwards, certain things have to happen in order to develop what's called contraction, yeah? Now imagine that before actually myosin head binds to actin, it has some ATP attached to it. And this position is called detached position because yet the myosin head is not attached to actin, yeah? Afterwards, when calcium arrives into the cell, and afterwards I'm going to describe how calcium actually arrives into the cell, how its level presses into the cell. When calcium arrives, then it can bind to troponin C, and tropomyosin is getting ready to move to another direction and free these binding areas for the myosin head. But before that occurs, the myosin head actually hydrolyzes the connected ATP. The ATP, when it's hydrolyzed, turns into ADP and phosphate. And now, since the myosin head already attached ADP and phosphate, this is called cocked structure, which is absolutely ready to attach to to actin once its binding sites become free, yeah? Afterward, when the binding sites are already free because of attachment of calcium to troponin C, the myosin head attaches to the actin binding sites. And now afterwards, this is called already the cross-bridge structure. And afterwards, when the phosphate detaches from this cross-bridge structure, we get what's called power stroke, when the myosin head is now able to change its angle and to slide, which actually leads to contraction. And thus, the overlapping of the myosin and actin happens, and we get actual contraction, yeah? Specifically, this occurs only after the detachment of the phosphate group and only after the detachment of the ADP from the myosin head and attachment of new ATP to the myosin head, we get once again a resting position, our detached position, and now the muscle is relaxed. And now it's ready for new muscle contraction to occur with the exact same circle, of course. Okay, so first was detached position, the resting position with the binding of ATP, then the cocked position after the hydrolysis of ATP, then the cross bridge once the myosin head actually attaches to the actin molecules, then detachment of phosphate group leads us to power stroke and actual contraction when the angle actually changes. And finally, when ATP is detached and new ATP is attached, we get once again resting detached position, okay? So this slide is just this slide actually zooms in the whole process, which I have already described, so I'm just skipping it. And as a final sum up, let me remind you that two types of filaments, like thin filament and thick filaments, take part in this whole process of skeletal muscle contraction. And this process is thin filament controlled and mediated. Why? Because the troponin actually and tropomyosin, these two thin filament molecules, control the movement and contraction of the skeletal muscle, because the tropomyosin covers the binding grooves, and if the calcium is not attached to troponin C at first, and if tropomyosin is not removed from this binding group, then no type of contraction actually happens. That's why we call skeletal muscle contraction thin filament mediated. Thick filaments are myosins. Myosins bind to ATP, and well, they hydrolyze the ATP, then they can actually change their angle and lead to contraction. Yeah, and finally we get myosin power stroke, which I have already shown to you. And then once again, I'm repeating the most important detail on this slide is that these processes being filament controlled and mediated. Yeah. Okay. Now let's move on to motor end plate. Well, I mentioned that actually neurons control the muscle contraction, somatic neuronal system, and this is done by releasing acetylcholine into the neuromuscular synapse, where the acetylcholine attaches to its own receptors on the postsynaptic membrane and some area of the postsynaptic membrane on the sarcolemma. And sarcolemma is the membrane, plasma membrane of the muscle cell. Yeah, you remember. And the specific area of postsynaptic membrane is called motor end plate. Well, the motor end plate has the folds, crests, which are rich of acetylcholine receptors. And when the acetylcholine is attached to its target receptor, then the voltage-gated sodium channels become opened. This leads to depolarization. And now let's see how the other steps actually occur. So here is our terminal synapse. I mean the terminal part of the axon, the synapse afterwards. So acetylcholine neurotransmitters called acetylcholines are released. They can attach to their target receptors. And here is our fold called T tubule located inside the sarcolemma. Which has a lot of channels on it. And when the depolarization phase actually reaches this specific area of the sarcolemma, a lot of channels actually can activate on it. Here we have some dihydropyridine receptors. What are these dihydropyridine receptors? They actually are proteins that span gaps between the T tubules and sarcoplasmic reticulum. This is our sarcoplasmic reticulum, where calcium is stored, and it is similar to endoplasmic reticulum, but it's specifically for calcium storage, and it's called sarcoplasmic in the muscle tissue, specifically muscle cells. So the dihydropyridine receptors are actually located between the T tubules and the sarcoplasmic reticulum, and they are also called L-type calcium channels, and they are capable of conducting some calcium. Yeah, but the main mechanism of muscle contraction and the main mechanism of increasing the amount of calcium inside the cell is not these L-type calcium channel, but the ryanodine receptors, which are located in the sarcoplasmic reticulum. And when these dihydropyridine receptors are actually activated because depolarization has reached the T tubule, when they change their conformation, this is a signal for ryanodine receptors on the sarcoplasmic reticulum to release some calcium from the sarcoplasmic reticulum into the cytosol, yeah, into the sarcoplasm. When the calcium actually is increased inside the cell, it can now easily attach to troponin C. Then you already know what happens. Tropomyosin moves from the binding sites, then the myosin attaches to the actin, and then contraction happens. Okay, so basically we started the discussion of what happens in sarcolemma. I mean, not sarcolemma, sarcomere. And then we described how calcium regulates this whole process. And finally, we described how actually calcium level can increase inside the cytosol. Yeah. And this is a sum up slide. This slide actually sums up the whole process, and this flow chart actually shows what happens when the acetylcholine is released. Then the sodium channels are activated and opened. Depolarization stimulus occurs, and this leads to action potential. Then the action potential is spread to the T tubules and open. Then some calcium channels, the dihydropyridine calcium channels, L-type calcium channels are opened. Also, this leads to ryanodine receptor activation and calcium release from the sarcoplasmic reticulum. Then the calcium level increases in the sarcoplasm, in the cytosol of the muscle cell. And finally, when it binds to troponin, it leads to contraction by aiding the process of myosin head and actin attachment. Yeah. Okay. And finally, I have to mention another channel, which is also located in the sarcoplasmic reticulum, but it's responsible for not moving calcium from this sarcoplasmic reticulum into the cell, but on the contrary, for moving calcium from the cytosol into the sarcoplasmic membrane. Why? Because when the contraction occurs afterwards, when it's already done and the relaxation has to be developed, calcium, this high amount of calcium has to go back to the sarcoplasmic reticulum. This is done by SERCA, our endoplasmic reticulum calcium ATPase. Here D is missing, sorry, in the name. And there's a special thing here is that it's ATP dependent, which means that when calcium is got back to the sarcoplasmic reticulum, this process uses the ATP hydrolysis, okay? Now let's move on to some terminology, and talk about twitching and summation. When the muscle is actually stimulated with a single electric shock, I mean, like this single action potential, yeah, depolarization phase. When it's, of course, of sufficient voltage, it quickly contracts and relaxes. And this one contraction and relaxation is called twitch. When the twitches can actually summate, yeah. If the second electric shock is delivered before the muscle has had a chance to fully relax from the first twitch, then the second twitch can be added to this first twitch, and this can lead to what's called summation. Now let me show you this on the graph. Here is our first stimulus. When the stimulus comes, when the action potential starts, then you know that the calcium channels have to be opened, and calcium has to be increased inside the cell, and calcium has to attach to troponin and stuff like that. And this requires a little, but still some amount of time. And this is called the latent period. I mean, between the stimulus, actual stimulus, and between the actual muscle twitch, I mean the contraction, yeah? So here there is a very little latent period when this whole preparation processes have to be occurred, yeah? Afterward, we get one twitch. But now imagine, if first stimulus has occurred and one first twitch has started, but the muscle has not relaxed fully yet. This is somewhere at this position. Well, it's relaxing. It has started relaxation, but is not really relaxed fully yet. When the second stimulus comes during this process, these two twitches actually can summate. Yeah, these two twitches can be added to each other, and we can get what's called summation. Okay. Okay. And another term is some gradient contraction, which means that if we actually increase the stimulus voltage, this voltage increasement can increase the frequency of action potentials, and of course the amount of calcium into the in the sarcoplasm. I mean, in the cytosol of the muscle cell. Yeah. Increasing the stimulus voltage can also activate more muscle fibers, recruiting them into the contraction. And this allows variation in the strength of contraction, which is termed as graded contraction, to be produced in response to electric shocks in vitro. So graded contraction just simply means that the variation, which actually depends on the stimulus voltage. Yeah. And now let's move on to tetanus. Let me show you this on the graph. When we have really a lot of stimuli, step by step, one by one, occurring really quickly, and twitches are followed, actually, there is very little relaxation time between them. Then these twitches can actually lead to some kind of not very smooth, but very jerky appearance of muscle contraction. And this is called incomplete tetanus. Well, if we increase the amount of stimuli, I mean, if we let the stimuli to become more frequent, so the relaxation times become shorter and shorter between the twitches, then this whole contraction becomes smoother, but still, this is an incomplete tetanus. We get the complete tetanus only once the relaxation time actually between the twitches actually disappears, and we get the whole sustained smooth contraction. And when it continues, when a lot of frequent stimuli are coming and inducing the contractions to be very smooth and sustained, then afterwards, after a certain amount of time, the muscle gradually loses its ability to maintain contraction. It fatigues, and once again, we get these jerky contractions, and their amplitude, of course, decreases. This is represented, this actually represents what's called fatigue of the muscle after tetanus. But the tetanus, this term is quite different from the disease tetanus, which is mediated by the bacterium called Clostridium tetani. So don't get confused. These are two separate terms, I mean, two separate words used for separate reasons, okay? Afterwards, now I want to describe what is force generated during contraction, yeah? Well, actually, in order for muscle fibers to shorten, when they contract, they must generate a force that is greater than the opposing force that acts to prevent movement of the muscles insertion, okay? Well, for example, when we lift a weight by flexing our elbow, the force produced by the elbow by the biceps brachii specifically has to be greater than the force, force of gravity in order to actually lift this specific cargo or specific subject, yeah? Now, let me move on to some types of contractions. Well, I've mentioned that some force has to be generated, but these forces actually differ in different types of contractions. Well, in the beginning of the lecture, I mentioned that in some cases, the joint angle can be changed. Well, we may get some flexion movements or extension movements, but in both cases, the the muscles are really producing some tension, yeah? The tension is always produced, no matter it's flexion or extension, but the actual length of the muscle changes. In flexion, it's shorter, in extension, it's longer, yeah? That's why these specific types like extension and flexion movements are called isotonic movements. Whereas if there is a sustained contraction of the muscle, if we are like trying to maintain our posture, or we are trying to hold a certain thing, for example, a glass of water in our hand, and doesn't even we don't even move our hands. I mean, neither flex or nor extend our muscles. Then we get what's called isometric contraction, which means muscle doesn't shorten, but the tension increases. Let me show you this with the help of the video. Let me explain this with the help of a little video here. Well, here is our isotonic contraction and isometric contraction. In isotonic contraction, we actually use some flexion and extension movements, yeah? But tonicity is always, tension is always produced by the muscle. But in isometric contraction, let me move to this isometric contraction. Now the muscle length is not really changed, but the tension is actually produced, yeah? This is the major difference between the isotonic and isometric contraction. Once again, I'm repeating, in isometric contraction like this, the tension is produced, but the length of the muscle is not changed. But in isotonic contraction, we actually change the lengths of our muscles. Sometimes they become longer, sometimes they become shorter according to extension and flexion, of course. but the tension is always produced. That's why it's called isotonic, yeah? Okay. Just a second, something is wrong with it. Yeah, okay. As for the clinical correlation, here I want to mention rigor mortis. What is rigor mortis? This is the stiffening of the body that begins a couple of hours after death and lasts for about 2 days, depending on the temperature and other stuff, yeah? Why? Because when the muscles are contracted, afterward, relaxation has to occur, and calcium has to go back to sarcoplasmic reticulum with the help of SERCA, yeah? And if you remember, SERCA, specific channel, which is located in the sarcoplasmic reticulum, is ATP dependent. Now imagine that after death, no energy is produced in the body, and no ATP is produced in the body. So SERCA is not really working properly, which means that calcium amount stays high inside the cytoplasm. Calcium stays attached to troponin, and thus the tropomyosin is not inhibiting the actin and myosin binding sites on actin, which means the cross bridges are still formed. And that's why muscles cannot be really relaxed after contraction. And this is actually called rigor mortis, which means that after death, muscles, none of the muscles in our body can be actually relaxed. That's why they are really rigid. And that actually happens because of lack of ATP, because of increased amount of calcium inside the cell, and still attached myosin heads to the actin molecules. That's because the cross bridges are still formed and muscle is no more able to relax, okay? And afterwards, now I can move on to the next topic, which is a bit different from the skeletal muscle contraction. And let me now mention some smooth muscles. Well, you know that smooth muscles are mostly located in our organs. That's why they are sometimes called visceral muscles. For example, those muscles which are arranged in circular layers are located in the wall of the blood vessels and bronchioles, which are small air passages in our lungs. Also, both circular and longitudinal smooth muscle layers are located in our digestive tract, in the ureters, in the ductus deferens, which transports some sperm cells, and the uterine tubes, which transports some ova. And alternate of contraction of the circular and longitudinal smooth muscle layers in the intestines, for example, produce what's called peristaltic waves, which are really special and important for digestion, which propel the content of these tubes in one direction. And generally, smooth muscles have different types, sorry, not types, but different functions in our whole organism and body. And smooth muscles are controlled not by us directly, but our autonomic nervous system. Okay. Well, smooth muscles actually do not contain exact same sarcomers as we described and discussed in the case of skeletal muscles, but they also do contain some amount of actin and myosin, and exactly actin and myosin attachment plays a role in smooth muscle contraction, but still the contraction mechanism is quite different from the skeletal muscle. Before I talk about the mechanism itself, let me tell you that there are some single and multi-unit smooth muscles. Well, smooth muscles are often grouped in these two functional categories. For example, let me start with the single unit smooth muscle. In case of single unit smooth muscle, the individual smooth muscles are electrically joined by gap junctions between them. These are our gap junctions, which means that when one smooth muscle is activated, the impulses can be sent to another smooth muscle really quickly, then to another smooth muscle. So they work as whole group and the whole syncytium, and they can contract really together. And these types of single units are also found in, for example, in the digestive tract, in the uterus, because the contraction has to occur simultaneously. And gap junctions, by the way, are also found in cardiac muscles. And in cardiac muscle, gap junctions are really important because the ventricles and atria of the heart have to really contract at the very same time. If that doesn't happen, then the arrhythmias can develop, which means the irregular rhythm can be developed inside the heart, and which is actually life-threatening and really dangerous. So gap junctions play a role in cardiac muscle as well. But now we are talking about the smooth muscle, yeah? The second type is multi-unit smooth muscle, which means that the axons of the autonomic nervous system have some varicosities, as you see here. These are our varicosities, little varicosities, which release neurotransmitters, and then that forms synapses on the patient with the smooth muscle cells. So here we have no gap junctions. I mean, directly they are controlled by the varicosities of these axons, which directly release some neurotransmitters, which then, of course, activate your target receptors on the smooth muscles, okay? Okay, now let me move on to the actual contraction mechanism of smooth muscle. The smooth muscle contraction is thick filament regulated in contrast with skeletal muscle. Why? Because here, we have actin and myosin, both of them represented in the smooth muscle, but myosin is divided. You know that myosin has some heavy and light chains, and specifically myosin light chains, which are the representatives of thick filaments, not thin filaments, are the modifiers of the actual smooth muscle contraction. That's why we say that smooth muscle contraction is thick filament regulated and mediated, but skeletal muscle contraction was thin filament regulated because their troponin and tropomyosin were those regulators. But here, myosin light chain is the major one, yeah? Well, in order for the myosin light chain to start this whole contraction process, phosphorylation of myosin light chain has to be occurred. Yeah, MLC actually stands for myosin light chain, so don't get confused. Now there are two enzymes called myosin light chain kinase, which leads to phosphorylation of the myosin light chain, and there is a myosin light chain phosphatase, which leads to dephosphorylation of the myosin. Yeah, and as I've mentioned, only phosphorylated MLC or myosin light chain can start contraction, but dephosphorylation actually leads to relaxation, the opposite process, okay? Now let's say what actually leads to this whole phosphorylation thing. Turns out that calcium plays a huge role in smooth muscle contraction, similarly to the skeletal muscle contraction, yeah? In skeletal muscle, it's used to contact and attach the troponin, yeah? But here in smooth muscle, we don't have any kind of troponin. We have calmodulin, specifically located in the cytosol of the cell, in the sarcoplasm. And when calcium actually binds to calmodulin and calcium calmodulin complex is produced, this activates myosin light chain kinase, which is, as you probably remember, responsible for phosphorylation of myosin light chain. And once the myosin light chain is phosphorylated, this leads to contraction, okay? Now let me see the whole stuff and whole thing on this very nice graph. Imagine that here is our axon. Some gradient depolarization has occurred there. The depolarization wave has reached the motor end plate, and now some voltage-gated calcium channels become opened. These calcium chains are, by the way, also called dihydropyridine calcium chains. Not chains, sorry, channels. Exactly the same name of the receptors which we are found in the skeletal muscle. Dihydropyridine receptors, yeah. And when these voltage-gated calcium channels open and calcium enters the cell, it contacts with calmodulin, then the calmodulin calcium complex is produced, which brings myosin light chain kinase from inactive to active state. Once it becomes active, now it's able to attach some phosphate group to myosin light chain, and this is called phosphorylation. And this can lead to cross-bridge formation and contraction. Once the contraction occurs, then the myosin phosphatase has to deactivate myosin light chain, to detach phosphate from it. Afterward, cross-bridge inactivation occurs, and this leads to relaxation, And this is whole loop, whole circle. Once new calcium arrives and new calcium modeling complex is produced, then once again MLCK becomes active, then the phosphorylation of the MLC occurs, and this leads to contraction, and afterwards again relaxation, and so on and so on. This is the main idea. Oh, sorry. And finally, let me mention that calcium, it has to be increased inside the cell, but this is mediated by two receptors. First one is the L-type calcium channel, which is our voltage-gated calcium channel, and which is also called dihydropyridine receptor. And another one, which is also responsible for increasing calcium level inside the cell, is the receptor located in the sarcoplasmic reticulum, which is responsible for release of calcium from the sarcoplasmic reticulum, where it is specifically stored during relaxation, and during contraction, it has to be released from the SR. Okay, so in exactly the same way as in case of skeletal muscle, here we have two types of main receptors, one located on the sarcoplasmic membrane, one located on the sarcoplasmic reticulum. And finally, the final slide just summarizes that the two major regulation mechanisms are in the smooth muscle cell, and these are the calcium level inside the cell and myosin light chain phosphorylation, dephosphorylation process. Okay. And finally, this is a clinical correlation. We have some calcium channel blocker drugs which can block and specifically attach to some voltage-gated calcium channels. You remember I mentioned these L-type calcium channels called dihydropyridine receptors located in the membrane. And imagine if they are blocked, then no calcium can enter the cell, and calcium modeling complex cannot be produced, and phosphorylation processes cannot be occurred, which means this can lead to smooth muscle relaxation. And this is necessary for some vasodilation. Why? Because sometimes when there is a hypertension in a patient, vasoconstriction actually occurs in the patient, which means the smooth muscles inside the vessels are contracted, and this leads to a decreased radius of the vessel. And this leads to high blood pressure in the person, But if we want to decrease the pressure, we want to dilate the vessels, we want to increase the radius of the vessels, and we can do this by relaxation of smooth muscles. In order to relax the smooth muscles, we can block these voltage-gated calcium channels there, with the help of some calcium channel blocker drugs. They will lead to relaxation of the smooth muscle. Relaxation can lead to vasodilation, and these can finally actually decrease the blood pressure. So these drugs are actually quite helpful in management of hypertension, especially in old people, And finally, at the end of the presentation, you see your homework, your book, the chapter, and the parts of this specific parts of the chapter, because you don't have the whole chapter actually. So thank you for listening. Hopefully you enjoyed it, and see you during the next seminar. Goodbye.